Welcome back to BioMindscape. Today we are going to know about some important diagrams of NCERT, that is incomplete dominance, codominance. So first we will see in this chapter, that is in the law of dominance, we can see example of heterozygous, that is capital W and small w, where one allele it masks completely the other. For example, the test cross with violet flowers. This is an example given in NCERT where the violet flower is completely dominant over the white. If the unknown flower is homozygous dominant, that is capital W and capital W, then all the offspring will be violet. And if the unknown flower is heterozygous, that is capital W small w, there is a chance of 50% violet and 50% white. And this is classic Mendelian, where 3:1 or 1:1 ratio is obtained. Then second diagram, it is incomplete dominance. That is a blending that is given, like if we cross a red flower with white flower, we will get a pink flower, that is red homozygous, capital R homozygous. If we cross with the white one, smaller homozygous, then we will get the F1 generation, that is all the flowers will be pink in color, that is capital R and small r. So why the color that is pink we get? Because the red allele, that is capital R, they produce some pigment that are not enough to make the flower fully red, while the white allele produce no pigment. So the capital R small r, they produce intermediate amount of pigment which give a mixture of red and white color, that is a pink color. Then F2 generation, when the pink plant is self-pollinated, the phenotypic ratio that we get is 1:2:1. Okay, that is 1 red, 2 pink and 1 white. The genotypic ratio that we get is 1 capital R, 2 capital R small r and 1 small r, same as that of the monohybrid cross. But always remember here the ratio is 1:2:1. In complete dominance, here what happens, the heterozygous look different from both the parent, not like the dominant one, and it is a blend. So this shows, the figure shows the red, pink and white snapdragon example of flower. Then codominance, where both the allele fully express. There is no mixture here in the codominance, like in blood groups. Both allele in the heterozygous are fully expressed at the same time. No masking of other, and there is no intermediating blend, that is mixture. So there is a gene like I for a blood group. It has 3 alleles, IA and IB and small I. These are multiple alleles and more than 2 alleles for a particular one gene. Like capital I A. A will produce an A sugar antigen on the red blood cell, and B will produce B sugar antigen of red blood cell, and small I will have no sugar production. So codominance between the IA and IB, here we can see the genotype is having a blood group of IA and IB if blood group is AB. So both A and B sugar are produced and present on the RBC surface, that is the blood cell surface. You can see both the A and B antigen clearly. There is no mixture of the antigens. Why it's codominance and not incomplete dominance? We have seen that in incomplete dominance, the red and white if mix, we get a pink color, and in codominance both the trait appear fully, that is A plus B will give a AB blood group, while both the antigen are visible. Multiple alleles, even though each person has only one allele, one from the mother, one from the father, the gene is the population that has 3 possibilities, like IA, IB and small I. So this create a 4 blood group types, like A blood group, B blood group, AB blood group and O blood group, with 6 possible genotype. Now these are the genotypes that are given for particular blood groups like AB, A, B and O. So these are the O will have small I, whereas the AB will have capital IA and capital IB, and for genotype A and B it is given. One more example of incomplete dominance which is mentioned in NCERT is starch synthesis in pea seeds, where the round and wrinkle but with size variation, like capital B are the large grain starch grain which have round seed, and small b are small starch grain with wrinkle seed, and capital B and small b is intermediate starch grain. So today we get to understand complete dominance, incomplete dominance, and codominance. So complete dominance where 1 wins completely, that the ratio is 3:1. Incomplete dominance is a mixture, like red plus white will give pink, 1:2:1. Then codominance, both win equally, means both traits are seen, that is codominance. You can also learn that Snapdragon pink incomplete. ABO blood group is codominance plus multiple allele. Now let's know about the monohybrid cross, Punnett square and test cross. So today we are going to know about the monohybrid cross. So in this, the pure tall capital T homozygous dominant, pure dwarf small t homozygous recessive, and hybrid tall, that is capital T small t heterozygous. We have crossed them. Then the parental generation that we have crossed a tall into the dwarf, that is we have crossed the tall capital T into the dwarf one. So the gamete that we get, tall plant gives only capital T, whereas dwarf plant gives only small t. So the F1 generation, that is capital T small t, that is heterozygous. So we can see all the tall plants there because T is dominant and it shows the law of dominance. So F1 for a self pollination, if we do a self pollination for F1, we will get capital T small t into capital T small t. So Punnett square, if we use a Punnett square, so we get this genotype, that is 1:2:1, that is 1 tall, 2 tall heterozygous and 1 dwarfed, that is 1 small t small t. If we see the phenotype, that is 3 tall and 1 dwarf, that is capital T plus capital T small t. Then the phenotypic ratio we get 3:1, whereas genotypic ratio we get 1:2:1. So this is the famous Mendelian ratio, 3:1 ratio. And here important thing is that the F1 generation consists of tall plants, but 2/3 are actually heterozygous, that is capital T small t, and 1/3 are homozygous, capital T. And what is a test cross? Test cross is a special type of cross where the unknown genotype, which are dominant phenotype plants, like the unknown plant is capital T, and it shows a tallness. So this tall homozygous will be crossed with the small t recessive parent, and this cross will tell us the genotypic conformation. Like capital T small t, that is 100% tall, we will get, that is unknown plant capital T homozygous tall will be crossed with the small t, and we will get all the offspring capital T small t, that is 100% tall. Again we will take this capital T small t heterozygous tall, we will cross it with small t, so the offspring we will get 50% of tall capital T small t plus 50% of small t dwarfed. So this was the case 2. By the result we can get that if all the offspring are tall, then the unknown tallness will be shown, and if the half tallness are given there, so the half dwarfness will be given. So unknown tall, that is capital T small t, will be there. It is used to test the genotype. Like violet flower, we have crossed this flower and we get this genotype. Like violet flower are dominant, we have crossed violet flower with the white flower, which shows the same principle. And in law of segregation, at the time of meiosis, the alleles are getting segregated. Here the gametes consist of an allele, only one allele. So the F2 genotypic ratio we get is 1:2:1, and phenotype we get 3:1 in the monohybrid cross. the Punnett square. In that we can easily calculate the possible zygotes. In test cross we may get a ratio of 1:1, that is 1 tall and 1 dwarf, if they are heterozygous. Then cross type monohybrid test cross and the second test cross where half tall and half dwarf we get. So in monohybrid cross we get 3:1. In test cross capital T is crossed with small t we get all tall, and if we have test cross capital T small t into small t half tall and half dwarf, that is 1:1 ratio we get. So for knowing such concepts, follow BioMindscape.